Today I'm going to present a comparative study of the vitellogenesis in Echinodermata, lessons from the sea star by these people. So for introduction, they discuss that yolk proteins are the main source of nourishment for developing embryos of oviparous animals. Vitellogenin, or VTG, is a major precursor of egg yolk proteins and is a female-specific which provides amino acids, phosphates, lipids, and carbohydrates to the embryo. VTG belongs to a large lipid transfer (LT) protein LTP superfamily. Vitellogenesis, also known as yolk deposition, is a process of yolk formation via nutrients being deposited to the oocyte or female germ cell involved in reproduction. Also, it precursors of the main yolk protein in Echinozoa, which is an example of sea urchins, and Holoperidea, they is an example of sea cucumbers. They are a member of the Clasperin superfamily, known as the major yolk protein or MYP. As compared to the BTG, MYP is not female specific, so it is both found in both sexes. Nevertheless, MYP has been implicated as a protein precursor found in asteroid sea star, which is the Parvularia exigua or P. exigua. BTG is heterosynthetic, means that it is synthesized outside the ovary. Furthermore, MYP's transcripts have been found in the gonads of both sexes, and also it is a major swimming fluid in sea urchin and sea cucumber. Yolk protein precursor proteins in sea star are synthesized in pylorixica, and based on the RT-PCR, BTG transcript in asteroid these exigua that I mentioned earlier were recorded ovary and pylorixica. So the aim of the study that they discussed in the introduction were to characterize the genes encoding, to determine the distribution and the abundance of respective transcripts in the tissue and cell types, and to identify the primary yolk protein in the sea star. As we move on, I'm gonna explain about the materials and methods that they use. So the animal sampling dissection, they only selected sea stars that only have more than 30 millimeters of their the arm length, which the pylorixica and gonads were removed, and the removed ones are collected and washed with filtered seawater. And they are also used for C2 hybridization, and coelomocytes are collected through suction by inserting a needle into the overall surface of one of the sea star that we are always doing when we're getting coelomocytes. And they also did the isolation and sequencing of cDNAs, including PRBTGs and PRMYP. The total RNA was isolated from ovary or pylorixica, and the RNA concentrations in DNA treated samples were quantified with spectrometer, and the total RNA was used for synthesis of single-stranded cDNA. And forward and reverse primers were designed to amplify the entire expected open reading frame sequence, and the PCR1 was amplified with an expected 5 kilobase pairs band which were extracted from the gel, and the PCR2 was performed to generate a high amount of DNA from the PCR antigen. And the reverse and forward primers were designed on the both concaves, encoding PRBTG2 to amplify the entire expected open reading frame sequence merging both concaves. And the PCR replica was performed for 35 cycles. And they extracted the amplicons for PRBTG1 and PRMP were ligated into PGMPEC vector, which is a system for the cloning. And plasmids are isolated, was loaded in 0.1 and 8% agarose gels. And the plasmid sequence was performed using specific primers, and they call it as primer whopping in order to obtain the full length of cDNA sequence of the clone target. So an example of primer whopping is like this. It's like that one. Oh, it's not showing? Oh, never mind. It's not showing. Sorry. And they also did for phylogenetic reconstruction to see if they have similarities within the family. So they analyzed and performed using the PHYML program, and they edited using the Mega version 5. And for the STS page, they use the specimens of regularis were spawned via intracellular injection of methyl adenine. And the ova were transferred and washed through several changes of filtered seawater. Embryos and larvae were collected at the several stages. And the collected embryos were washed again with filtered seawater at least 2 times and was centrifuged at 10,000 grams and stored at 70 degrees Fahrenheit. And a bullet blender was used to homogenize the ovarian tissue of related eggs embryo in a homogenizing buffer. And the supernatants were used for protein assay. The STS phase was performed using 5% gel. And the protein extracts from the selected tissue were separated by STS phage. It was stained with Comasi brilliant blue, and a marker was used to compare. So the several stages were identified in this reproduction cycle, which recovery stage, growing stage, mature stage, partly spawn stage, and span stage. For each one, the stage 1, which is the recovery stage, it is the abundance of young pear-shaped oocytes surrounded by somatic cells, individual follicle cells. And the growing stage is the pear-shaped growing oocytes, where the most abundant germ cell type. Somatic cells other than follicle cells decrease in number. And in stage 3, the mature stage, which is fully developed oocytes, were most abundant in ovary. And the stage 4, which is the partly spawn stage, the ovarian lumen appeared empty as oocytes were spawned. So the growing oocytes and young oocytes lining the ovary wall were abundant, and the somatic cell number is increased. And the span stage is the ovaries individual few patriotic oocytes dividing oogo and increase in number. Somatic cells were abundant in stage 5. They also did the quantification PCR. The total RNA was extracted and DNA treated except for pseudocytes. So I will discuss this part why they didn't extract pseudocytes as the same to the total RNA in the discussion part. And that real-time quantitative PCR primers were designed based on isolated sequences for VTGs and MYT. And they did also the in situ hybridization. Plasmid containing vitellogenin 1 and 2 or MYP cDNA insert was subjected to PCR to generate partials cDNA using primers designed based on target sequences, and a small portion of the resulting probe was used for gel electrophysics to determine the synthesized probe length and integrity. And the results that from the study that they did was their identification of 2 novel VTGs in C star. So the 2 transcript encoding vitellogenin-like proteins were identified and selected as per VTG1 and 2. Using a primer walking, the cDNA coding per VTG1 was cloned and sequenced. It showed that 99% identity of the total length of the isolated per VTG1 cDNA in 4,901 base pairs to its corresponding config. The PCR opticon in PRVTG2 was sequenced and aligned with the corresponding config, yielding over 99% identity. The PRVTG2 opticon contained a 5,682 base pairs ORF that encoded a predicted protein composed of 1893 amino acids. So this is the example of the primer walking that they did. As you can see, the bold italics, which is this first 15, are the predicted signal peptide, and the stop codon is in asterisk, which is here. This is the start and stop codon. And the green highlighted ones are the peptides from band 1, and the yellow ones are the peptides from band 2, and this little one that are in that black background are the subtlelism cleavage site. So basically, it is also the same with this one, which is the difference is that these blue ones are the nucleotide sequence, and that's basically the difference from one. And the results from the C stars vitellogenin belong to the LTP superfamily and contain vitellogenin conserved domains. A lipoprotein N-terminal domain was found at the N-terminal end of both pure VTGs and frequent flows in the LLT module. This is the LLT module. The domain of the unknown function was identified in the middle region of the sequences, and the C-terminal of both PRVTGs was found. So, as comparing to the PRVTG1, this one and PRVTG2, they have 80, they have 32% similarity, and from the RPTG1, they have 88% similarity with the C star, which is this one, the C star, and this is the VTG1, and 42% with the nematode, 23% with the zebrafish, and 28% with the cloudy cut shark. And for the PRBTG2, this one, as we compared it to the nematode, it has 35% similarity, 29% with the Pacific oyster, 32% with the zebrafish, and 26% with the frog. We also did the SDS page. So these 4 bands are the prominent bands, and this band 1 has 260 base KDA. It matches with this amino acid sequences of PRBTG1 and 2. The second band has 660 KDA, which match with this amino acid of PRBTG1, and the band 3 has 80 KDA, which matches to the PRBTG2. So none of these 4 bands match with the clone part of the MYP from the species of P regularis, which is the C star. And these one are the tissue distribution of each PRBTG1, PRBTG2, and the MYP of the mRNA. So we can see that the PRBTGs and PRMYP were detected in all examined organs from samples from feet, skin, stomach, ovary, pyloric and coelomocyte. And the pyloric showed by far the highest in mRNA. As you can see, it is maybe the highest in all of the proteins. And you can see that, and the PRBT and the low transcript levels prevail in tube and feet, which is in this one. They are very low. They're literally very low in this protein. And the highest level of pure weight transcript, as to compare to this 2, is very high. See, the coelomocytes here is very high as compared to here it's not. And these are the mRNA abundance. So the highest PRBTG1 transcript levels found in the stages, which is in the stent ovary, and lowest in stage 3 is in the matured stage. So this one, PRBTG1, stage 1, stage 2. This is stage 1, 2, 3. They're very low. And there's a twofold increase in levels of PRBTG1 mRNA between stages 4 and 5. 4 and 5. They increased as compared to the first 3. And there's no significant difference in ovarian PRBTG2 or PRMYP, these 2. Across the reproductive cycle in CFP, that's said. And this B, D, and F, these 3. The pyloric transcript levels of PRBTGs and PRMYP change in comparable patterns. See, as this has a low, it significantly increased, then it becomes low, high, low, low, high, low. It's like a pattern. So it dropped to minimum levels of stage 5 and remained low in the recovery phase, which is the stage So the mRNA levels were at least 7-fold higher in stage 4 compared to stage 5. This is the in situ hybridization result. So these arrows are the positive hybridization to PRBTG1 into mRNA cells, so the somatic cells as they're pointing it to. In PRBTG1 here in stages 2 and 5, there is a strong signal at stage 5, which is the spent ovaries, these 3. And stage 2 signals were detected, but their intensities are lower than stage 5. So this one has lower intensities as compared to stage 5. And in PRBTG2, it is in ovary and in pyloric CCA signal. So somatic cells in stage 2, this one, as you can see, the arrows. And the probes are revealed in the ovary or in the follicles, in the follicle cells in this tree. And the strongest signals, which is in stage 2, ovary, and stage 4, pyloric cecum. So this ovary and this pyloric cecum. So the pyloric ceca in the pyloric ceca here is weaker as compared to stage 2 in PRBTG1. And lastly, for the PRMYP, it was detected in the oocyte. The strongest signal, which is in stage 2 here, these 3, and the weak signals were also detected at stages 1 and 4, but I don't know why they told it, but it's not in here. In contrast to PRBTG1 and 2, PRMYP signals were detected using this method in pyloric ceca, though they use a different method in this one. In discussion, it is said that MYP has been reported to constitute the precursor of the primary yolk proteins in Echinozoa, which is the sea urchin. It is unclear that whether BTG is the primary precursor for yolk proteins in asteroids and whether the sea stars produce an MYP or not. BTG is the main precursor for yolk protein asteroids and not MYP. Asteroids are the sea stars. MYP transcripts are not detectable in the surrounding somatic cells but rather in oocytes, so they are detected in oocyte instead in somatic cells, as we can see from this one. Also, the findings strongly suggest that BTG is the primary protein destined storage in the yolk oocytes and subsequent utilization by the developed asteroid embryo. PRBTGs belong to the BTG protein clade in phylogenetic analysis. I want to show you this, but it's not showing in the presentation, so I'll just show it later. And both PRBTGs and RBTG proteins are belong to the LLTG superfamily. The P regularis, which is the C star, the expression of the PRBTG1 was reported in ovary and pyloric ceca using the reverse transcriptase PCR. And both PRBTG1 and 2 of P regularis detected its transcript in various tissues, which is in this one. And the presence of PRBTG transcript in coelomocytes highlights their likely contribution to vitellogenesis, and it is possible that secretory cells of the digestive epithelium produce in both PRBTGs. In conclusion, they conclude that the 2 BTG genes with their PRBTG1 and 2 are detected mainly in the pyloric ceca and ovaries of the C star P regularis, and proteins encoded by these genes are quantitatively the dominant proteins, the ovum star of the C star. And close associations exist between PDG transcript abundance and the progression of the reproductive cycle, which are the 5 stages that I mentioned earlier. And PDG is not prominent role protein of the C star P regularly and not MYP. So basically, the major protein is found in sea cucumbers. Also, both PRBTG are detected. Oh, wait, that's not. Professor, I want to show them the picture that the clade. I don't know how it looks like.